Hello.
Good afternoon, sir.
Hello, everyone.
Hello.
Dr. Malviya here.
Hello, sir. Hello.
आनंद भाई कैसे हैं?
मैं ठीक हूँ। बड़े पुराने लोगों से मुलाकात होगी।
बड़े स्मार्ट लग रहे हो यार।
अरे अभी इस उम्र में और क्या लगेंगे। जब 80 पार कर जाते हैं तो बड़े स्मार्ट ही लगते हैं। हेलो, मेहरा साहब भी हैं, सीता जी भी हैं।
I know.
I haven't reached 80 yet, sir.
नहीं, you देखो, you are the second generation. I am in
No,
towards,
sir. Very much your generation. Just a few years. Not
I'm
too
more
much.
towards GP तलवार।
ये साउंड क्यों नहीं ठीक आ रही? बीच में कट रहा है।
क्या मेरी साउंड नहीं आ रही ठीक?
नहीं सर, मुझे ठीक आ रही है।
मुझे भी ठीक आ रही है।
तो मेरा जी का ही गड़बड़ है।
हाँ सर, ट्रैवल कर रहे हैं जी। सर इज इन जयपुर।
ओह, ऑब्वियसली रास्ते में कैसे सुनेंगे?
यार मैं जयपुर में हूँ।
बहुत अच्छी बात है। इट्स अ नाइस प्लेस टू बी।
हाँ नहीं, अभी आया। एक हमारी कॉन्फ्रेंस है यहाँ। The immunology has grown so much. Now we have the Immuno-Oncology Society of India.
Why not? There should be autoimmune society also.
तो
There is some echo somewhere.
हमारे जमाने में you remember आनंद, वो होती थी वो छींक छांक की सोसाइटी एक।
Allergy and applied immunology.
Allergy and applied immunology कहते थे उसको।
I think that has died. That has kind of died.
I don't think it is alive anymore.
नहीं, उसके वो करता थे, उसका क्या नाम क्या था यार?
शिवपुरी।
क्या था वो करता जो थे?
डॉक्टर शिवपुरी।
हाँ, शिवपुरी।
He is no more, unfortunately.
Yeah. अच्छा, he's no more.
It got merged with the TV.
Right.
नहीं,
Kanga, welcome. How are you?
Good afternoon, everyone, sir, madam. Namaskar.
Good afternoon, everyone.
Good to see you all. Happy New Year.
Everyone, Happy New Year. Whenever you say, I can start.
Sure. Dr. Deepankar?
Yes.
Where is he? Where is Deepankar?
Should I start, Taruna?
Yeah. Let me request Dr. Deepankar Nandi, professor at Indian Institute of Sciences, Bangalore, to take over the session from me. Yeah.
All right. So it's a great pleasure for me to introduce Professor Sita Naik to chair the session. Professor Naik is one of the former presidents of the Indian Immunology Society. She was also the former head Department of Immunology and Dean.
Deepankar, just give me a moment. I just asked the IT guy to... Hemant, can you please show Dr. Sita Naik's slides?
Yes.
Please. Sorry, Deepankar. Sorry.
Yeah, no problem.
Yeah.
सीता को सारे जानते हैं यार, उसको बोलने दो।
हाँ जी।
अरे भाई, बहुत से लोगों ने ज्वाइन किया है। हमारी रूमेटोलॉजी पूरी सोसाइटी ज्वाइन कर रही है भाई।
नहीं, पर अगर हिंदुस्तान में लोग सीता को नहीं जानेंगे तो कहाँ जानेंगे?
नए बच्चों को जानने दीजिए।
We are all
many implications.
Here, there are a lot of young people. They don't know any of us. Please believe that.
Yes.
You might think everybody knows you, but they certainly don't know most of us because they are much younger.
Sound का issue है कि
generations down the line, यार.
सर, आपके पास कुछ इशू है सर, आप रीजॉइन कर लो सर।
हाँ, मेरा तो बड़ा क्लियर आ रहा है साउंड सबका।
एको है कहीं पे।
आपके पास कुछ इशू है सर, आप रीजॉइन कर लो। मेरा तो बड़ा क्लियर आ रहा है।
Good morning, madam.
Deepankar, you can start, Deepankar.
Yeah, fine. I was just waiting for your... okay, let me start over again. So it's a great pleasure for me to introduce Professor Sita Naik, who was one of the former presidents of the Indian Immunology Society. She was also the former head Department of Immunology and Dean Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. She was also a former member of the Board of Governors, MCI. Professor Naik has a long list. So what I will do is I will only select parts of it because so that we can move on with the talk. She established the immunology laboratory at the GS Medical College in Mumbai and provided immunology services to the KEM hospital. Also, at the newly established SGPI in Lucknow, she conceptualized, planned, and supervised one of the best departments in immunology in the country. So, under her leadership, SGPI really grew up with a specialty program in immunology. All right. So she has supervised several PhDs and MSc scholars, and as a member of the board of council of MCI, she coordinated and developed an undergraduate training program which was implemented by MCI in 2018. As you can understand, she served on various committees of ICMR and several other departments. I request Professor Naik to now take over and introduce the speaker. Thank you.
Thank you, Dipankar. Can I have the slides in Dr. Malviya also? I think there were slides.
Yes, ma'am. Hemant, please display Professor Malviya's slides. Thank you.
Right. Anyway,
full screen. Yeah.
In Dr. Malviya's case, he is one of the doyens of immunology in India. And I think I met him first in December 1973 in the first meeting of the Indian Immunology Society. So it's almost, that makes it 50 years around this time. If I remember, I think it was in November or December of 73 that we had that first meeting in the biochemistry department of All India Institute of Medical Sciences. So, and of course, as Narinder had mentioned, as Dr. Malviya had mentioned, also had close association with over the years because he was a very close friend and classmate of my own mentor, Dr. Shobha Saigal of PGI Chandigarh. So he was a founder member. He was a former president of the society. He is a very active clinical rheumatologist, for immunologists, it's often easy to forget that he is actually a clinician because his involvement with immunology has been phenomenal. So he's been very active in the Rheumatology Society, where he is the godfather of rheumatology in India. He's like everybody's guru there. I had the pleasure to be at Indore this year for the meeting after a few years at the rheumatology conference, and his status there is something else. It is to be seen, to be believed. He has had a number of areas of expertise in rheumatology, the clinical aspects. Can I have the next slide, please? He has trained a huge number of students at All India Institute of Medical Sciences. And although, unfortunately, AIIMS never sort of made a proper, didn't get into the area of establishing a degree in clinical rheumatology during his time. I mean, in the formal sense that we say students from his department, we don't have those kinds, but a large number of our rheumatologists practicing around the country have been trained by him. He has over 400 research papers, and he is widely known globally for his work in rheumatology and immunology. He's a recipient of a large number of awards, including the Shakuntala Amichan Kamala Mohan Award of the ICMR, Boots Oration Award and Lifetime Achievement Award of the Indian Rheumatology Association, a number of other awards of the IRA, including the M.N. Passy Award. And I think the list, he has condensed the list himself in a very modest manner, because otherwise it would have been very long. It's a pleasure indeed, sir, to chair the session where you'll be presenting your work. So if you can invite.
Thank you, Sita. Are you? Am I?
Before you speak, I'm sorry. Where is Narinder wanted to also say a few words before you spoke.
Yes, I specifically requested Taruna to give me some time in the beginning, because then I have to go for the conference. And how can I not speak? A person with whom I had years and decades of association. And Rekha and Anand used to walk around in the campus of the AIMS, and everybody thought that was a very handsome couple. But 1973 was again a year which brought us together, because that was the year when Bruton for the first time demonstrated HLA association, HLA B27 association with ankylosing spondylitis. And at that time, there was no understanding of the broad spectrum of SSA, as we called later on, the seronegative spondyloarthritis. But his passion, I then started establishing the HLA services in AIMS. And this was one clinician who would come from the third floor down into the lab as well. So we used to have such great interaction. I would go to his OPD and he would come down into the lab. And I think that's the kind of basic and the clinical collaboration we want. So much so that I learned so much about the various diseases that he used to diagnose, and we published extensively. I think our papers, probably the number is about 60 to 70 over there, trained so many students as well. And one other passion that Anand had was teaching, education, and which he does even now. Even now, he's rheumatology, and I think those are probably fourth or fifth or sixth generation rheumatologists, and he sits with them like a young boy and sort of takes lectures for them. That's the passion that he has. I think that's what keeps him going. That's what keeps him very active. And of course, he is very fond of exercises. Exercise is the one thing that he will not leave. And the other thing that he's very fond of is singing, music. Music is first love. Cricket also is a very good love. So I think he keeps himself is a complete human being. I see in him a very renowned rheumatologist. Of course, you can say the number one rheumatologist of India. And no wonder that the international organizations have given him the title of master. He is the real master in his subject. And all due to the fact that he kept teaching all the time. And I think this is a lesson for our youngsters. Teach as much as you can, because when you teach, you know your deficiencies as well. You learn so much. You read all the time. And he has that habit of getting up in the morning and read and read. So Anand, I think they couldn't have found a better speaker than you to be the speaker of the Golden Jubilee year, because you have been also, like us, the founder member of the IIS. I do remember those days in 70s, early 70s, late 70s, when you would come from the third floor, because you were on the third floor, Dr. Talwar's department. I would come from the floor number 1 and up over there. People used to all the central point was the Department of Biochemistry there. And that's where the birth of the Indian Immunology Society was done. And you have been associated with it right from day one. Thank you so much for agreeing to give this talk. And we all are looking forward to it.
Am I audible?
Yes, sir.
Yes, you are.
But my, I think the video is not coming, right?
No, sir, we can't see you.
Yes.
Something has gone wrong. Can somebody help me? How to?
पहले तो आ रहा था.
हाँ, पता video. Oh, okay. Now it will.
Stop video वाला button उसको आप.
Start video. Now
जी.
Just let me fix the whole thing properly. Okay, right. Now let me share my slides. Okay. Professor Sita Nayak and Professor Narinder Mehra, I am overwhelmed, really. Honestly, I am overwhelmed. I am not that great or something. I'm just an ordinary person who has tried my best during my career to be updated in my field and do as well as it could be. But I will definitely like to thank you all. And it's such a pleasure to meet many of my old colleagues on webinar this afternoon. And a special thanks to Aruna from BHU. And you know my connection with BHU, my great-grandfather's university. He created it. And it's indeed an honor to be invited in a talk being organized by that university's Department of Immunology. And I'm thankful to Karuna and her colleagues for inviting me for this talk. Now, anti-nuclear antibody evolving understanding over the years is the talk that I will be giving. And the reason for it is because I'm, as you have been already told, I'm a physician, clinician, and my interest is in the field of now a group of diseases that have been renamed and now called anti-nuclear antibody associated diseases. The old name was connective tissue diseases, and in the middle, American College and the European Association of Rheumatology, they gave it the name of systemic immunoinflammatory rheumatic diseases. That's also a very good name. But generally, it is called systemic connective tissue disease, and CTD is the common name. But I will insist that these are diseases associated with anti-nuclear antibodies, and the group I will be naming. But here I will also like to mention, anyway, my disclosures, none financially, and the paper that I have read in detail for preparing for this lecture is given there. These are some additional key references that I have used in my today's talk. And there are some that have been highlighted with a pink reddish color. They are my colleagues, as well as some of them have been my mentors and close friends, and especially Dr. Meroni, who keeps coming to India, visits India quite frequently, and still continues to teach us and guide us on how to interpret anti-nuclear antibodies. And of course, David Pisetsky and Lipsky are the famous names in the field of anti-nuclear antibody in today's field. And they are leading the whole field and telling us how to interpret these tests and how to not incorrectly interpret and how to correctly interpret those tests. So with that clarification, I would like to clarify one more little point. Anti-nuclear antibody is the title I gave. But what I meant was anti-cell antibody. And this difference I will not be making during my talk. I will be using these two terms interchangeably. Historically, it was anti-nuclear. But when further studies were carried out, it was quite clear that we are not only talking about the epitopes in the antigens of nuclear material, but we are also looking at the whole cell and all the structures within the cell and the antigenic epitopes within the cell. So now, recently, it has been proposed that instead of anti-nuclear, we should start calling it anti-cell antibodies. But you know the problem, if we change the keyword, then millions of papers will not be available if you are writing a review. And that's why the name anti-nuclear still continues. But what we mean is anti-cell antibody. Now, these antibodies are seen in a large group of closely related conditions that I already mentioned. One important point to mention right in the beginning is that these molecules against which we are trying to see the presence of antibody in patients, they are highly conserved molecules, and they are components of all the mammalian cells. And there are hundreds of them. So when we say anti-nuclear or anti-cell antibody, we are not after only a single antibody. Actually, this is a group of antigens against which the antibodies are seen. And this group number is increasing over time. So I will like to make it simplified this afternoon for your understanding and try to make it as simple as possible. Can we make a simple screening test? And that's the first step. There are about 500 epitopes and molecules in the cell against which the autoantibodies can be created, can be seen in diseases. Can we make a single simple screening test that will detect irrespective of what is the specificity of that antibody and how to go about it. Are anti-cell antibodies present in this person or not? That will be the question. The patient comes to me. I take the history, examine the patient. I feel this is a patient in that group of ANA positive antibody group of diseases. Can I order a single test that will tell me yes present, no present? This is what my lecture is going to be. And then in the end, I will tell you something more about those number of increasing number of those epitopes in the cell against which antibodies are being found. I will start with one little slide of historical importance, the horror autotoxicus by Paul Ehrlich. In late 19th century and early 20th century, he was the biggest and the most famous name in immunology, and he propounded all what we understand about the clonal selection. Actually, he didn't describe, but he almost gave the whole impression, and he gave the theory about it by his hand-drawn slides, which are legendary. But there was a problem. He did not believe in autoimmunity, and he insisted there cannot be autoimmune diseases because that will be an antithesis to survival. If there is an antibody against self, that will not be compatible with life. And therefore, he said it is impossible to make autoantibodies, and thus his obstinacy in this area delayed the recognition of autoimmune diseases by at least 4 decades, which was a sad story because during those 4 decades, many hundreds and thousands of patients with autoimmune diseases were neither recognized nor looked after nor treated. And that's the tragedy of that. Then, however, in the ensuing years after that, convincing proof that there is formation of autoantibodies in diseases. And these are some of the references. I will not go in detail, but I will just like to make sure, look at Hargrave's paper, and that is 1948. When we describe Hargrave's work in the next slide, this was the seed sowed for the detection of anti-nuclear antibodies. How? Like this. To cut the long story short of this famous physician at the Mayo Clinic in Rochester, Minnesota, USA, in the year 1948, he published his article. He was treating many patients with systemic lupus in those days. In short, what he did was he took this serum sample from the patients with systemic lupus, incubated them with healthy bone marrow derived from normal volunteers, incubated them, and then looked at the bone marrow cells. What he found was that in the serum there was some factor which was causing damage in polymorphonuclear leukocytes. And you can see in the slide right there, these two cells are dying polymorphonuclear leukocytes. And the nucleus is getting extruded in a dead form. And polymorphs being the phagocytic cells, they immediately ingested the dead nucleus, giving a shape which is visible like this. Hargraves gave the name LE cell phenomena because he was able to repeatedly demonstrate this type of phenomena using this serum from patients of systemic lupus and utilizing the normal bone marrow cells from volunteers to produce these cells. And he said it has to be some serum factor in the patient's blood and the patient suffering from systemic lupus. Now, once it was known that there seems to be something which antibody that is reacting with nucleus, and nucleus is in the cell. How to demonstrate an antibody against antigen in situ in a tissue? Till then, it was, a gel diffusion test, hemagglutination test, complement fixation test. All of them was in fluid phase. So it was easy. But how to demonstrate the presence of an antigen in situ in a whole tissue? like nucleus. And that's when they utilized the now famous, of course, immunofluorescence study that conveniently was described from Harvard Medical School Department of Pathology by Albert Coons. And I was rather lucky to have met him when I was in training in Boston.
इतकं सांगून पाठवलं की हे परत नीट कर आणि त्याला काहीतरी टेबलला काहीतरी नावगाव दे ना.
Sorry, there is some disturbance. Somebody is speaking. Anyway, so Kuhn's technique of technique of labeling an antibody and using it as a guide to show the antigens against which that antibody is reacting with became very popular in those days. And this was then taken up by Holbrook, a Britisher, and he happened to be my teacher for 6 weeks in Lausanne, Switzerland in 1971, when I attended the course How to Perform Anti-Nuclear Test. And he said that one of the best substrate will be murine liver, because liver is a well-defined big organ. We can cut sections. The cells are big enough, and the nucleus is quite well-defined. So if we use murine tissue to provide us the substrate for anti-nuclear antibody, that will be very ideal. So here is the substrate, cryostat sections cut, put on the slide. Patient serum, suspected lupus patient serum, put on this. If there is presence of anti-nuclear antibodies, they will combine with the nuclear substance in the tissue of liver of murine origin. This reactivity will occur. Then we can wash off the extraneous proteins from the serum of the patient. Only the antibodies against nucleus will stick. Then we add the second antibody, anti-gamma globulins, that would be labeled with fluorescein. That then will be put under microscope, fluorescent microscope, and they should give a yellow, a beautiful apple green fluorescence. And right there you can see this is All India Institute first ANA that we did, and that's why it is a faded slide. Instead of apple green, you can see the nucleus in light yellow coloration, but you can imagine it to be apple green in color. And we found that to be an excellent screening test, that is murine hepatocytes, cryopreserved section, to be used for detecting by indirect immunofluorescence test of Holbrook as one of the most suitable tests for the detection of and screening for anti-nuclear antibody. So antigen substrate, that is cryopreserved murine liver. Patient serum is put on top of that, then washed off. If the antibody is present, it will stick to the nucleus. Fluorescein conjugated antibody against human immunoglobulins, then incubated, then washed off and examined under UV light microscopy. Look at this. The fluorescent microscope has shown now clearly these are the cells which are shining apple green, and so the patient serum would be reported as positive for anti-nuclear antibody test. And this is the more detailed murine liver right there. Put the patient serum, then wash it off, put the conjugate, wash it off, and see under the microscope. And you will see these cells, and the nucleus will show the apple green fluorescence. Not only that, you can often in those days, even in the murine liver cells, you would be able to give the pattern of staining. And we used to describe at least minimum of three patterns: the homogeneous, the speckled, and the peripheral pattern of anti-nuclear positivity. Life was simple in 1970s, 1980s, and early 1990s, and everything was fine. I just digress. In New York, in those days when Hargraves described the Ellie cell, and later many laboratories around the world, including Holbrook in London, described the anti-nuclear antibody, Henry Kunkel's laboratory in New York, the famous immunologist, I don't have to give the details, he was of a great stature, almost like many others of those days. His student, Eng Tan, he told Eng Tan, and Eng Tan is still going strong at 94, just like our colleague, Dr. Professor G.P. Talwar. Eng Tan is also in his 90s and going strong, publishing papers. Kunkel told Eng Tan when he was a young man, these autoantibodies against anti-nuclear positive, what are the antigens against which they are reacting? So, Mr. Eng, you go ahead in the laboratory and try to establish what antigen they are reacting with. And what Eng Tan did was, he also had many patients for lupus. One of them was called Mrs. Smith, who was a painter, and she has painted her own back looking at a mirror and presented it to Dr. Eng Tan, who used to be my mentor. I used to meet him to learn about how to do ENAs, and he taught me how to do that. So this is Dr. Eng Tan. He says that I will take cell extract. And what is the best place to get the cells from? Fetal thymus. So bovine fetal thymus he took, made extract, saline extract out of that, and using gel diffusion technique. And here in the central well, he put the cell extract of thymus, and in the wells around, he put the patient sample. And here is the sample which he put from his patient, Mrs. Smith. And your overnight incubation showed a precipitin line right there. And this was the SM antigen. Name was given because Mrs. Smith had this antibody that reacted with antigen in the cell extract, which was then named SM. And there were many other similar names in a patient, La, in a patient RNP, etc. So now these names became very popular. Of course, now we have more defined name, ribonucleoprotein and etc. So we have now the second stream. One was the screening for anti-nuclear antibody, but the second stream took it the specificity of the antibody. What portion of the nucleus or cell, which epitope or which antigen in the cell and nucleus the antibody is reacting with. I will leave it at this stage because my topic is not to give you the details of these ENAs, but to go with more detail of anti-nuclear antibody screening test, the immunofluorescence test, and where are we in the present day. So, using the murine liver as the substrate and homemade reagents and home assembled equipments, as you can see here, along with the help of my colleagues. Narinder Mehra was already there speaking about me, and my colleague Dr. Bhuya, Dr. Professor Ramesh Kumar, unfortunately he is no more with me, and my technical team, Ramesh Taneja, who is unfortunately not with me, and Gopal Singh and Sobe Singh, all the technicians. These are the equipment we used, homemade plexiglass made in my All India Institute's workshop. We collected patients from our patients with myeloma, microglobulinemia, IgG, IgM, and IgA myelomas. And Abbott and Mar immunochemistry, the book we will take, purify these antigens, inject it in the purify them and inject in the goats of our animal house in All India Institute and raise monoclonal antibody, sorry, not monoclonal, monospecific antibodies against gamma globulins, then stain them with, label them with fluorescein, and using our own home laboratory cast mice, we will remove the liver. Dr. Bhuya will make the cryostat, and using a monocular epifluorescent microscope given to us by our director, Professor Ramalinga Swami, we would start doing we started doing anti-nuclear antibody tests. Here we are. The first anti-nuclear antibody test done between 1970 and 73. And these are the original slides from the laboratory where I established this test. And with the establishment of the test, the so-called ELI cell became history, and it was not used anymore. And the anti-nuclear antibody fluorescence method became the drug, the test of choice for the screening for anti-nuclear antibodies. We also established many more antibodies, but I will not take your time for that. Anti-thyroid, anti-gastric parietal cell, anti-cytoplasmic nuclear antibodies, etc. Now we come to the name that we have given. I already told you the original name was the diseases where these antibodies are seen, anti-nuclear or anti-cell antibodies, connective tissue diseases. Then came the name systemic autoimmune rheumatic diseases, and the present name is anti-ANA associated or anti-cell antibody associated diseases. These are the diseases in which systemic lupus is the leading. This is the disease we are specializing in. And of course, we say rheumatoid and systemic sclerosis and other diseases where ANA are positive. But we will take the prototype disease systemic lupus and explain a few more points about this test. So, the prototype disease and systemic lupus ANA, where screening test done by immunofluorescence will come positive, will give a positive result, and we will say, yes, the disease that we diagnose clinically systemic lupus has been confirmed. Then we will set up the specificity testing tests using cell extracts in gel diffusion, and now, of course, with the ELISA and even more advanced tests like bead testing, etc. And we found that in SLE, the specific antibodies which reacting with the nuclear material are against double-stranded DNA as well as against SM antigens. Then autoantibodies, especially in SLE, can be relevant for confirmation, suspected diagnosis, classification, as well as for monitoring disease activity, because DNA antibodies go up and down along with the activity of the disease. If the disease is going in remission, these antibodies go down in amount, and they start rising the moment patient is in danger of a relapse. Thus, cryopreserved murine liver served us for the next 2 or 3 decades. It was a simple test. It was quite insensitive, however. And insensitivity actually was not bad, because if it is insensitive, it was giving us only real positives. So no false positive and no false negative. How beautiful. So we were naive. We kept on using that till the whole area was muddled by advances in the field of testing of ANA. And I will take up this. But before I go to that, this a very crude test that we set up in All India Institute on murine liver gave us enough information, and we wrote papers after papers on systemic lupus and its prevalence in population, and you see the results on these papers. But then came the issue that made the whole field very complicated. And again, it had to come from the master, and that is Entan again. In 1978, he says, murine liver is not a very standard tissue. It varies from laboratory to laboratory, species to species. Some people are using rat, others are using mice. The homemade fluorescein conjugate has no standard. Somewhere it is very poorly prepared, somewhere it is very good. So the results that we are obtaining is very variable. It is not standardized. During that time, a particular cell line derived from human laryngeal carcinoma was cultivated and cultured by a commercial laboratory, and it was patented, and they patented it, and they started propagating the cell, and this cell became HEP2 cultured cell, which today every immunologist uses in case they are working on any human cell line. This cell line was standardized, so Tan and Nakamura, they said, why not use the HEP2 cultured cell? These are big cells with very huge big nucleus where staining pattern can be studied very well. It will be a standardized substrate for tissue. And then these commercial laboratories also were very clever. They made the fluorescent double antibody also very specific. They prepared it in a very standard way. So standardized reagents became available to carry on and carry on the ANA test in each laboratory anywhere in the world, and the results will then be comparable. So uniformity came in practice by the use of HEP2 cells and the reagents that were being provided by the same laboratory that was providing the cells. Then what happened? it raised several issues. Mouse liver was not very sensitive. But the moment we shifted to Hep2 cells, it became a highly sensitive test. So many false positive results started coming. In addition, we were very naive when we were studying the fluorescent pattern on murine liver cells. Only 3 patterns, speckled and homogeneous and rim pattern. Sometimes we will also see nucleolar pattern. But 9, from those 3 or 4 patterns, the Hep2 nucleus was so well defined and so big, 29 different patterns have been described till today. So that was the other problem. Which pattern goes in favor of which disease? That also became an issue. And lastly, when the patients with systemic lupus, but the test became very easily available, they were being done all over the place in every clinical pathology laboratory in every city. And repeatedly the same patient will come, today it will be done and next week they will do it again, and 3 months later patient comes for a follow-up and the test will be repeated. So the follow-up result on anti-nuclear antibody test became available, and it became very clear that the test that was positive at the beginning when the patient came for the first time becomes negative in the follow-up. So if you are unlucky and you have seen the patient for the first time treated by some other doctor and you don't know what treatment was given, you might see this is a clear-cut patient with systemic lupus, and yet the test result is negative. You didn't realize that the patient was treated and some of the antibodies, they disappeared over a period of time. So the problem of false positivity and false negativity arose directly because of the ease of the test and the increased sensitivity of Hep2 cells, which were now being used as substrate for doing the anti-nuclear antibody test by immunofluorescence. And therefore was then an international consensus was set up, international consensus on ANA pattern initiative. A group came in picture. These scientists sat together to define different patterns, identify them, giving them the name, and they used the term A cell. A cell number 1, A cell number 2, A cell number 3. So now we know AC1 up to AC29 different patterns are described, and for each pattern it is available online. You can see that if it is pattern A3, AC3, then what are the diseases that are likely to show this pattern will be available online. So you can not only know that the nuclear antibody test is positive, but you will also know the pattern, which will indicate what is the likely diagnosis in that person. So now, and in addition, not only the nuclear pattern became visible, but using Hep2, even cytoplasm started showing some forms of immunofluorescence in certain patients. So it was clear that not only the patients have anti-nuclear antibodies, but they have antibodies against other components of the cell, and that's why the name came anti-cell antibodies, and some of these antigens were present actually in the cytoplasm. Rho is one of the antigens which is almost always present in only cytoplasm. And similarly, some of the antigens that showed a positive anti-nuclear test were actually identified to be present in mitotic bundles. So it became quite complicated and became a specialist area, not only to do the test, but also to tell us the pattern, the AC number, so that we clinician come to know what disease it is likely to be. And to make life simple, I have given this to my fellows. Normal is 7, 16, 23. AC2 is a unique thing. If it is present, the person is normal. Moreover, it projects that such a person who shows DFS 70 or AC2 pattern of immunofluorescence will never develop a connective tissue disease. That's very interesting. Then SLE 1, 4, 5 and these numbers, if this is the pattern of immunofluorescence on Hep2, then the patient is likely to have SLE, and so on and so forth. These numbers tell us what is the likely disease underlying from which the patient's blood has been drawn. But as I mentioned, increasing sensitivity of the test has created the problem of false positivity. And because the test is simple, patients keep on getting it done over a period of time. So those who were positive become negative, and the patient start wondering, do I have lupus? Patient may have been misdiagnosed lupus. So both the problem of false positivity and false negativity became a major issue once we started using HEP2. And then again came the master and Tan in 1997. He, along with some of the stalwarts, I mentioned the papers, including Smolin and many others, Manny and others, they all got together and they exchanged samples of patients with systemic lupus diagnosed by them, so definite cases of lupus. And they found that, and they also took a number of normals, confirmed normals from their population where they were working. Using these healthy normal controls and using definite positive samples from serum of samples from SLE patients, the test clearly showed that at a lower dilution, low titers of anti-nuclear antibodies are present in normal individuals in up to a third of them. So anti-nuclear antibody then was realized is present in normal healthy persons. But high titers, if you dilute it up to 1 in 160 titer, then only 5% false positives will be seen and 5% false negative will be seen. And thus, this group of workers led by Tan made it an international guideline that when you are reporting on anti-nuclear antibodies using HEP2 as the substrate for ANA reporting, you should use a titer of dilution of 1 in 160 or more to indicate that the test is positive. And in addition, of course, you will be describing the AC pattern, the fluorescence pattern on HEP2 cell nucleus. Thus, Lipsky and Pisetsky and Lipsky recently made it known to the whole world that 30% of normal population have anti-nuclear antibody, but these are present in low titers and they are not detected in up to 95%, and only in about 5% if you dilute the serum to a level of 1 in 160 or more. Then they went in great detail why a test is giving negative and why a test is giving positive. The patient has been given treatment. The patient has been going on for a very long time. Maybe the disease is under remission. Maybe there is involvement of kidney, and all the antibodies are flowing out in urine. So the amount of antibodies has gone down in the blood, and so on and so forth. And I won't go in detail. So it became clear that positivity and negativity depends upon the type of patient you take, type of person you are studying, and the dilution of the serum that you are using to give a positive or a negative result. So similarly, in drug trials, it was found out that many of the patients we diagnosed as lupus, and the drug trials were conducted and it showed a beautiful result. 30% of them were ANA negative. Even lupus patients showed negative, but when they went in detail, they found that these were the chronic patients going on with treatment, steroids and immunosuppressive drugs. So their antibody levels had gone down after years. And then it became very clear that if you take only fresh cases, untreated, newly diagnosed cases of lupus, the positivity of ANA at a high titer is present in near 100% cases. And that brought in the statisticians. And that is where now clinical pathology laboratories are recommended to give the result as follows. Now, all of us will have to just go through it in a very short half a minute. I will explain. Imagine this is the outpatients of one of the rheumatology clinics. In my OPD, there are 100 patients sitting in this morning at 9 a.m. Of them, the diagnosis of lupus is carried by 10 of them. These are relatively new case. In 90 of them, the diagnosis is something else. They are lupus or Sjogren syndrome or myositis or whatever. So 90 of them is non-lupus, 10 of them are lupus, and anti-nuclear antibody tests were ordered, and the laboratory only diluted it to the strength of 1 in 80 at the highest titer that they were using to report the test. Using 1 in 80 titer, they found 9 of the 10 lupus patients gave a positive and 1 gave a false negative. Out of the 90 patients, 13 gave a positive result, which will be clearly false positive because they were not patients with lupus. And 87 gave true negative because they were not patients with systemic lupus. And using the formula here, calculation for a ratio which is called likelihood ratio, which most of you already know, the positive likelihood ratio can be calculated based upon these figures to be 6.9. And negative likelihood ratio you all can calculate will come to 0.11. Let us go to the next. We change the dilution to from 1 in 80 to 1 in 160. Suddenly the numbers changed in this group. True positive and true negative of lupus patients remained the same because they had high titer positive. But the false positive numbers went down. And if you now calculate the positive likelihood ratio, from 6 it went up to 18. And the negative likelihood ratio from 0.11, it has now reached 0.1. Now, if you go to what is the cutoff ratio that we should believe to be a good cutoff for positive and good negative likelihood ratio cutoff, the numbers are these. Now, you have seen when we use the titer of 1 in 160 cutoff, we get much higher positive likelihood ratio of 18. So that was the excellent way of reporting, and all those reports which were sent as positive at that titer will be true patients with systemic lupus. And ideally, as we mentioned, less than or 0.1 as we achieved by using a titer of 1 in 160. We have now given them a solid report, definitive report. If this test is negative, patient is unlikely to have systemic lupus. And thus, today's clinical immunology laboratories are requested and recommended that please report not only the dilution at which you have given the result, you have diluted the serum up to what level, what dilution, and 1 in 160 being the recommended dilution at which you report positive and negative values. Along with that, you also say that Hep2 was the substrate that was used with the standard reagents provided by the same laboratory. And finally, you have also to give the likelihood ratio that this test will provide us when we take standard cases of SLE and standard cases of normal persons. If we do this, then the test gives wonderful and reliable reports. Now there comes the issue. Who orders the test? I gave you the example of my OPD or OPD from renowned rheumatologists who are well versed with the diseases, and they can clinically make a definitive diagnosis of these diseases. But now we take large number of other patients, other clinicians and physicians. For example, over a period of time from rheumatologists, now primary care, pulmonologists, oncologists, neurologists, cardiologists, dermatologists, gynecologists, all of them will start ordering, oh, I suspect this patient may have lupus. So indiscriminate ordering of the ANA has become the rule nowadays. And that has brought in the problem of pre-test probability, which has gone down drastically. And that has created problem compounded with more and more sensitive tests that are being used by different laboratories, including the solid phase assays, the immunoblots, multi-analyte technologies like there are many of them. I won't be going in detail. It's difficult for me even to pronounce them. But using more and more sensitive tests and patients not being screened clinically properly, we are getting into difficulty of large number of false positive tests that are being reported nowadays from clinical pathology laboratories. I will like to end my talk with one small little paper here. Nowadays, and this paper is not very old, 2003, in one of the major laboratories in USA, 232 samples were received for request for doing anti-nuclear antibody test. The laboratory knew about TAN's work and recommendations of the American and European League that the titers should be the serum dilution should be used up to 1 in 160 to give the report, and above that, if it is positive, you just report positive. But below that, you will report negative. What they found was only about 2.1% of the samples sent were actually from lupus patients, and all the rest of more than 98% of them were not lupus. What a waste of large effort of the laboratory and the resources of the laboratory when indiscriminate ordering of the antinuclear test is being done by those who have not screened clinically, and they have not been stringent in making a clear diagnosis, clinical diagnosis, making the pre-test probability as high. Unless the pre-test probability is high and only indiscriminate ordering is being done, then all the effort of the laboratory will be a total waste and waste of money and efforts of the nation. I would like now to end with a glimpse of the future, how this anti-nuclear or anti-cell antibodies are going to be screened for this group of diseases that I have been discussing. By the way, the scientists have been after the increasingly interested in seeing these anti-nuclear antibodies are reacting and anti-cell antibodies are reacting with what antigens? What are these antigens in nucleus and in cells and different components of cells? Till today, till now, more than 500 autoantigens present in cell and cell extract have been isolated and identified and reactivity established. I will not go in detail. You can read about them. It's all over the place. And with increasing automation, with particle-based multi-analyte technology being the latest introduced in the laboratory, and I'm told that within the next 6 months, most of the laboratories, I won't take the name, but some prominent laboratories have informed me that they are already in the stage that they are going to make multi-analyte technology as the main test in their laboratory. Imagine if we are not sending them the correctly selected patients with lupus or this group of disorders, they are going to face major problems of false positivity and false negativity. However, if we do it properly, it will help in early diagnosis, evaluation of prognosis and risk stratification, choosing a proper response, and then seeing whether the patient is responding or not. These are some of the newer technologies, difficult even to pronounce the name, Aliba, PMAT, etc. I'm told PMAT is the one which has been now taken over and been taken over by many of the laboratories, and this is going to be the standard with which anti-cell antibodies will be reported. So, with the use of artificial intelligence, large big data from clinical records, laboratory findings, and socioeconomic, because different races give different number of ANA positivity in their lupus. For example, a certain race, I won't name them, they give very high positivity, while in certain other races it has been shown that even in patients with lupus, they give a little lower number of ANA positivity. So that information, then transcriptome information, and then treatment, whether they were treated or not treated, all this information will be fed in the computers, and then artificial algorithms will decide what is the most likely diagnosis and when can we exclude the diagnosis and when can we make the diagnosis. And that seems to be the future of anti-cell antibody and anti-nuclear antibody. Take home message, therefore, use HEP2, and nowadays we call it HEP2000 because the Rho antigen has been transfected in the nuclear material. So now the Rho has become from cytoplasmic antigen to now become a nuclear antigen. So most of the laboratories are now using immunofluorescence, using HEP2000 cell as the substrate, using that as the substrate at a cutoff point of 1 in 160. The test will be reported. If it comes positive, automatically, reflexly, it should be sent for the second step for identifying which antibody is it reacting with, which extractable nuclear antibody this material is reacting with, and thus we will report the ENA specificity also along with the positivity of immunofluorescent anti-nuclear antibody. And thus comes the protocol, which is strongly recommended for all the clinical immunology pathology laboratories to report. They should report, and this is the good clinical practice they should be practicing. A positive result should use HEP2 or 2000 cell. The titer they should be reporting is cutoff point 1 in 160 for clinical work, 1 in 80 for research work. It should include the N titer up to 1 in 160 to start with and goes up to how many thousands. If possible, positive and negative likelihood ratios must be mentioned. Using that test, the positive likelihood ratio is 18 or 17 or 12 or whatever, and negative likelihood ratio is 0.01 or 0.1. And then what is the AC nomenclature? AC number is 3 or 4 or 10 or 29 or 23. And then also suggest what will be the next second step that a clinician should order to see the specificity of the ENAs, and which will then help further categorizing our patients with definitive diagnosis of whether the patient will fit in with lupus or with Sjogren syndrome or with myositis or with scleroderma or what have you. Such a report will add value to the laboratory reports, and it will help clinicians like me in diagnosis, prognosis, therapeutic decision making, and in deciding about the treatment and the drugs. Lastly, I will leave with this slide. We all immunologists have seen that if you repeat antinuclear antibody test in a patient with systemic lupus after a few weeks, you don't find any change. The same titers are present of different ENAs. You repeat it after 6 months, the titers don't change. You repeat it after 2 years, titers don't change. What changes is antibody against DNA. Double-stranded DNA antibody therefore have become the antibodies which we follow our patients with in the follow-up studies. Antinuclear immunofluorescence is never repeated because that will give us positive over a period of time. But anti-DNA antibody keeps on fluctuating with the status of the disease activity. If the disease is responding to treatment, antibodies go down. If the patient is relapsing and nearing relapse, the antibody will rise and we will get ready. Oh, the patient is coming with relapse. Let us restart the treatment. The issue is, what explains this? Is it possible that actually most of the ENAs are being synthesized and produced by long-lived B and plasma cells? Long-lived B cells, plasma cells hiding somewhere in the crevices of bone marrow and not being attacked by the treatment that we are using today. All the B cell therapies that we are using, including rituximab and others, they are not acting on long-lived plasma cells. And that's why the patient goes in remission, but the moment you stop the treatment, patient starts relapsing. Is it then possible that actually long-lived B cells, when they will be the treatment targets, we might achieve full remission in these patients? And that also may be possible that here anti-dsDNA antibodies are not being synthesized by long-lived but short-lived B and plasma cells. And that's why they keep on going down on treatment and coming up at the time of relapse. My hypothesis, and it is being talked about, there is a recent paper that looks like that we are dealing with long-lived and short-lived plasma cells making anti-cell antibodies of different varieties and different types. This is a subject which I leave you, my colleagues, in basic immunological research to help us with. I hope it made sense, and I reached the end of my talk. Thank you for your kind attention.
So thank you, Dr. Malviya. And you'll all agree that was a masterclass. Very few of nothing has actually been left out regarding the evolution, history, the personal journey through this, what seems really very simple, but which is not such a simple test actually. And Dr. Malviya has highlighted all the problems that go with it. and how crucial it is actually for the management of these patients, that we have a good test. And in some ways, these tests were not so sensitive and so globally standardized in the days gone by. But I think in the earlier days, Dr. Malviya, when there was a good laboratory clinician interaction, in some ways, I think the patients were more benefited. Now there is so much of confusion and there is so much of dissociation between clinicians and the laboratories that sometimes I personally feel that something is lost in that lack of dialogue rather than a dialogue.
So right, you are.
Yeah. So if anybody has any questions, I know there are a number of people who are listening to this. I see. If anybody has any questions, we
Ma'am,
have. Yeah.
this is Kalpana here. Can I ask a quick question?
Yeah, please do.
Yeah. Sir, this is Kalpana from Department of Biochemistry, AIMS, New Delhi. It was a pleasure to hear your talk.
My old institution.
Sir, sir, you very rightly mentioned that the memory B cells may be, residing in the niches of the bone marrow, and therefore the antinuclear antibodies may not be able to bind and target them. But during any kind of disease activation, maybe during the activated stage, these antibodies, if given, would be more effective. Just as if you draw a parallel in the case of viral latency. So when you reactivate the latent cells, which will start producing the pathogen, and then you use an antibody, then it is more effective. So, so what is your comment on that? Like about
Let me clarify. I think there is slight bit of confusion. What I said was that there are these anti-cell antibodies seem to be of two distinct variety. One being produced possibly, I mean this is hypothesis, possibly by long-lived plasma cells. And so long-lived plasma cells are till today not being targeted by the therapies that are available. But there are some antibodies that are being produced by short-lived plasma cells like anti-DNA antibodies. What we have in our armamentarium for treatment is the rituximab type of drug. There are a few others which target these short-lived plasma cells. Once we use them, the antibody titers will go down. But what we are now trying to aim is to find out drugs which are targeting long-lived plasma cells. And I will give you the answer. The CAR T cells, which you all must have read recently, it was in New England Journal of Medicine, and now it has become a major way of treating cancers. CAR T cells will go and kill all the possible types of B cells, and the whole body will be repopulated by fresh normal B cells. And that will eliminate autoimmune diseases completely. So this is what my answer is to the question that was put in.
Thank you, sir. Thank you very much.
Sita, can I ask a question?
Welcome back, Narinder.
Yeah, please
no,
go
no.
ahead.
I was actually, Anand, I wanted to leave and go for the conference, but your lecture was so absorbing, I couldn't go.
And your photo was there.
I wanted to know, I mean, you listed a few factors, like who orders the test, the dilution factor, and the other things. Now, the PMAT test that has come up, I wanted to know, does it also make a difference whether the disease is at the early stage or is it in the advanced stage? Does the strength of the antibody vary from early to the more advanced disease?
In fact, Narinder, it's a wonderful question. This is exactly what I tried to mention, but the time is always a constraint, and I give this talk over a period of 3 hours. Still, it will not be complete. The issue is, here you are sitting in the OPD, a fresh case for the first time referred by somebody. It looks like lupus, and you take the history. The patient is absolutely a fresh case, has not been given any immunomodulated drug, has not received cortisone. The patient is you can say, naive to any treatment. This patient will have what natural lupus will show. It will have antibodies that are being produced by different type of B cells. It will have antibodies from long-lived plasma cells. It will have antibodies from short-lived plasma cells. And when you start doing the titers, you will get the real thing. High titers are present, and that's why they cut off of 1.1 to 160 or upwards. Then you have a patient who has been given Ayurveda, homeopathic alternative systems, and then you have a patient who has been given steroids over a period of time by all kind of physicians who have no idea what is the dose of steroid, what form of steroid to be given, and then they start giving immunomodulatory drugs in different varieties of them, and there are so many of them available, giving biological therapies even, not knowing the diagnosis. And then the patient is completely messed up. Some B cells are gone, some B cells are still alive, some T cells have disappeared, so helper cells are not there. What happens to those antibodies? Where are you? Remains a mystery in such cases. So when a messed up case of this type comes, whether PMAT gives any answer or not, will remain a big mystery till you first study pure, nicely diagnosed by those rheumatologists who know what lupus is, set up your laboratory, find out your false positivity and false negativity, taking some normals and real lupus patients, calculate the pre-test and post-test probability and likelihood ratio. Then you come up. And then these PMAT and what other tests, they will give you wonderful results. And in addition, they will give you a set of antibodies that if these set of ENAs are present, it is likely to be a subset of lupus where neuro involvement will be the main thing, or kidney involvement is going to be the main thing, or hematology is going to be the main thing. And therefore, you hit this molecule with using this monoclonal antibody as a therapy. That's what I see as future.
But I think
just one more small point. You are the master of autoimmunity. Can you tell me one other disease in autoimmunity where the antibody titer is so very high? Where here you're diluting 1:160. How about type 1 diabetes? Any other disease where the autoantibody titer becomes so high?
Actually, Sita will be the best one because she has studied much more of this. I just take the example of one of my diseases, that's rheumatoid arthritis, where titers are in thousands.
Diabetes is a different type of an autoimmune disease. If you talk about the rheumatic autoimmune diseases, I think there are cases as Sjogren's where we will have very high autoantibody disease, especially when they are not in remission and when they have severe disease. Some of the cytoplasmic, anti-cytoplasmic anti-neutrophil antibodies, which we have in very high titers. So there are, at least in rheumatic diseases, I agree that in organ-specific autoimmunity, anti-thyroid also now.
Very high titers, yeah, in thyroid also.
Titers. You get very high titers. I just wanted to comment to Dr. Malvia's issue of, what response to Narinder. I think the problem is partly because of these assimilated large laboratories and the setting up of these laboratories. These parameters, individual laboratories do not have to set up, false positive and false negative ratios and predictive values. They will not spend money testing a range of normals in their subpopulation and establish those for themselves. They just take it off the labels of the boxes. And so that does cause limitations.
Major problem.
The aggressive kind of medicine that, we are learning to, I don't know whether we are learning, but we are forced to live with in modern times. So those are compromises that you are making. It's unfortunate that even in academic institutions like All India Institute or where I work, the pressure of whatever of the system forces you to go from these kinds of things to the commercial entities. But I guess
I tell you something very interesting. There is a commercial laboratory in Delhi, very famous one. I won't name them. After hearing that I am giving this talk, They told me that they are interested in, and they might be on, because I gave them the link to come and join. They told me, sir, we will like to invite you to help us in guiding us how to make the positive predictive value and negative. I told them you will have to spend a lot of money and I will do the research. You give the money and I will provide everything else. Let us see whether they come back to me or not, because money, the moment you ask that you give money for research, they back out.
And I think another area which you touched upon in the last couple of slides, which is, well, I guess we are at the end of our career that way, is definitely artificial intelligence.
Oh, yes,
The
yes,
way
yes.
it has stepped into
Yeah,
radiology, people are
yeah.
actually talking about radiologists becoming redundant by 2030,
Yes.
because the AI is much better at reading a PET and a SPECT than human eyes.
Absolutely.
Yeah. Similarly, I don't know how it is going to go in laboratory medicine. All of biochemistry has become automated, but in these kinds of areas as well, as you are showing, I think the role of AI is something that's going to roll out. It's going to replace the test, and I don't know how quickly it will replace the man behind the microscope as well.
Yeah, I think don't get excited too much on the AI. You never know. Let the AI be in the field for some time, and then we will know. Human brain is a human brain.
Yes, Dr. Rao.
Can I ask one doubt? Anybody look after some of the markers on the B cell? Either on the B cell or T cells, either upregulation or downregulation of any of the specific markers.
Now here I will request Sita, because you see, I'm more a clinician rather than
What
reading.
markers are you talking about?
And because the B cell contains so many important markers that will trigger the release of antibodies actually. And any of them,
No,
any
I
novel
think,
markers.
yeah. That a lot of basic work has been done on the B cells in SLE, but here we are talking the diagnostic point of view, right? So we need to have the antibody already in the serum. Those are, in fact, that is a big issue in SLE, is that when does the start of the disease and when does it start? Because by the time we see the patient has symptoms and already has the antibodies. So obviously all this has started some months to years prior to the first contact with the doctor. So these are early events, very difficult to study them primarily because we don't know when the disease starts. We don't know the trigger.
And that brings up to another point, that there are relatives of the patients of lupus who show anti-nuclear antibody as positive. And whether they will develop lupus or not will depend upon the type of ENAs and the mixture of ENAs that they start showing at what stage. Just like rheumatoid. In rheumatoid also, the type of rheumatoid factor present before the disease appears has been now clarified. So these are the areas which are in future. Very soon we are going to be talking about them.
So, Anand, what is the algorithm in your opinion of diagnosing SLE? And you
Oh,
stated a
very
few
simple.
things. You
That is
stated a few
that
things,
is
and
what
one
I,
thing,
yeah.
one
That
thing you said that it should be ordered. The test should be ordered by a trained rheumatologist of the type
rheumatologist.
that you are, but you are only one.
No, there are. I mean, rheumatologists are getting trained very nicely in very many institutions, and they are excellent. There is no doubt. These young rheumatologists who are now DM or DNB or even fellows, they are no less. They can diagnose lupus, and they have reached a stage where I can say that you can rely on their diagnosis without much problem. Of course, there is always one here or there who may not be like that, but majority are very good, and you can depend upon them. There's no problem. The algorithm is very simple. You make a clinical diagnosis and you send the test for reporting and ask the laboratory to give them the N titer. If the titer is going 1 in 160 or above, you already suspect that there is a possibility of systemic lupus. You then ask them to do the ENAs. In India, because of the problem of money, issue of money, it is so expensive that we cannot order it to do it reflexly. Once we get the test result, then we decide what set of ENAs will be ordered. If I am feeling that this looks like lupus, I will just order NTDS DNA and NT Smith and RNP, and also do C3 and C4 levels. So we are rather miser in this area and select out ENAs, but in the Western countries, reflexly they will go for the whole series of ANAs, and their numbers are now rising to 500. And that pattern will then tell if this, and this is pattern, there is a scleroderma or any other disease. That is what I am doing, and that's what nowadays rheumatologists are doing.
So, Narinder, I don't think the issue is the lack. I mean, the issue is only when do they reach the rheumatologist. Unfortunately, since our referral systems
Yeah,
are very
no, but
robust,
Sita, and we
so
know,
there is a
and
delay between the onset of symptoms and the incubation with the primary, whoever doctors they see. Finally, they reach a rheumatologist. Already there is a
yeah,
time lag. That's
that's what they,
something healthcare has to take care of that. It's not
that's
something.
the
Absolutely
point.
right.
You know, in the 90s, I used to see, and I know quite a few those who now got diagnosed with SLE, but they said कभी जाते हैं तो कहते हैं SLE है, कभी जाते हैं तो कहते हैं नहीं है, तो हमें बड़ी प्रॉब्लम होती है. तो ये जो कभी जाने का कभी आने का इशू, अब ये hopefully it has
I
been
can
solved.
clarify that. You see,
हाँ,
these patients, they keep on doing their ANA by fluorescence. I told you, by
हाँ,
ANA will not help you. Don't do it after the first test that comes positive. You have to do ENA and DNA. That will go up and down. They keep on doing ANA. After 3 years, it becomes negative because the patient has kidney disease. They say, "अरे ये तो अब lupus नहीं है." What
हाँ,
nonsense!
वही
That's problem of the way our healthcare functions.
Yeah,
I think
yeah.
that's a larger issues. I mean, it's very difficult to tackle those at our level. We are talking about the science of things, what we see. Taruna, is if there are
We
no
have
more questions,
grossly
then
gone over the time.
that's okay.
No issue, sir. No issue. We have all
So if we have no
enjoyed.
more questions, then
Anand, just last point. New
Here we
Zealand is all
go.
out. Anand, the last point. New Zealand is all out for 108.
yeah, 108.
I was worrying, I haven't been
Unbelievable.
watching it.
Yeah, the first over was fantastic. Anyway, so
And
Taruna, so
Oh, so you all were cheating.
yeah.
can I
Uma,
ask a
no,
question?
no, no. I saw it before we started. It started 1:30 today. Yeah.
Oh, I was so nervous for this that I
Oh,
didn't see
nervous?
anything. I
You
was
nervous?
preparing.
That's
No, you
not
see,
possible.
I have colleagues who may not be clinicians,
So
and for them, I tried to make it as simple as possible.
thank you so much for asking me to chair this session. And may I now hand it over to Taruna or to Uma, I think, who will.
Yes, ma'am, I'm there. And this is our privilege to have you, ma'am and sir, and of course, Professor Mehra, that we really thoroughly enjoyed the session, gained so much of knowledge about the diseases that are associated with anti-nuclear antibodies, and so much of basic knowledge that is going to be laying foundation for many of our young students who have joined us today. And I would request Dr. Kaustuv Nayak from ICGEB to kindly present a formal word of thanks. Hemant,
Good
can you
evening,
please
everyone.
put on the slide?
Yes,
Word
sir.
of thanks slide. Thank you.
Good evening, everyone. Namaskar. First and foremost, on behalf of the Executive Committee of the Indian Immunology Society, with immense gratitude, we express sincere and wholehearted thanks to our eminent speaker, Senior Professor Anand Malviya, Head Department of Rheumatology, ISIC Super Specialty Hospital, and former President of the Society, for delivering the prestigious 4th IAS Golden Jubilee Lecture and sharing highly valuable insights from his clinical practice of more than 5 decades on autoimmune diseases. We are indeed honored by your enriching presence. We express our heartiest thanks to the chairperson of this event, Professor Sita Nayak, former head of the Department of Immunology, SGPGI, and former president IIS, for orchestrating this event in a scientific way. We thank all the participants across the country for their overwhelming response. Thank you one and all. We place on record our sincere thanks to the IT team, ICMR NIRCH Mumbai for their seamless work in making this event a grand success. Thank you one and all. Namaskar.
Thank you, everyone. Bye-bye.
Thank you, sir. Thank you so much. Thank you.
Namaskar, sir. Dr. Patil from Nimans.
How are you? Thank you.
I am fine, sir. Fine. Thank
Wonderful. So nice to meet all my old colleagues.
Sir, Uma,
Okay. Uma, kaise ho?
thank you so much, sir. And I wanted to say on this platform that being your student at AIMS, I also learned quite a lot. You and Mehra, sir, I was really fortunate to have you as my co-supervisor. And
Thank you
every
so
time I
much.
see you, sir, every time I learn from you.
Thank you.
Thank you
Thank
so
you so
much,
much.
sir. Thank you.
Thank you. And bye-bye, everyone.
Sir,
Yes.
there is a request from the audience, especially Dr. Reena Prajapati. She is requesting, can a session be held on interpretation and different techniques for ANA, different diagnostic techniques on various autoimmune diseases? So what is your suggestion in this regard?
I will request, just send it to me on my email or WhatsApp.
Sure,
Let me think about it, because if I commit without knowing what I am committing to,
Sure, sir.
I will first think about it. Do I
Sure.
have material and knowledge to give such a talk? If I feel that yes, I can handle it, surely I can do that. Only then I will commit myself. At this age of 84 years, it's not very easy to sit and start making slides, but I will try. But do send me what you actually want me to teach.
Sure, sir. We will do that. And I must share it with the audience that sir had such a hectic schedule throughout this month, and still he accommodated our request. And sir, it was a fantastic scientific treat for all of us. Thank you so much, sir.
Thank you so much. Okay, bye-bye.
Bye-bye.
Right, sir. Thanks.
Thank you, everyone. Thank you, all the audience. Grateful
Thank you.
that you all joined and made it a success.
Thanks,
Thank you very much.
Karuna, Bala.
Thank you.
Thank you, sir. Thank you.
In fact, being a student, Taruna, I was about to request you that can I say a vote of thanks? And then I saw Costa saying that, so I said it's okay. And then later on, because I really learned a lot. He was my co-supervisor, and it was such a great learning. Every time I would go to him, and he was. And also after he left AIMS, he did a study with us blindly. He never told me. He used to send samples, patients for testing. And then later he said, Uma, can you look at these referrals that I sent? And then we found such interesting data. We published one paper after that also. So he's been a great academician.
Absolutely.
50 साल हो गए.
And सबसे important बात है, so down to earth still, and still always ready to learn new things. At his age, I don't think so many of our oldies would like to even address the so-called Gen Next fields of machine learning and artificial intelligence. Even we are not so ready to accept them. And
even now, Taruna, in his clinic, he does many times the teaching sessions after the clinic, even at the private center. Like he said, chalo, everybody have lunch, and then during lunch we can have these meetings. So he really teaches and learns and discusses. I mean, it's amazing. It's really nice.
And Uma, interacting with him to organize this session was an amazing learning experience, because the way he responds, and if there is even one spelling misspelling is happening because of autocorrect, he will correct it and then write it again. And the way he has drafted his slides and disclaimers, and I've used the data from these papers, that is something to learn and adopt for all of us. We at times absolutely, we at times forget all these nuances which are really needed.
I was just about to write to you and Costa that can I? Then I said anyway there will be an opportunity, because in this platform we all have opportunity to still convey our regards and put up our questions. So I said it's fine, but it's so nice to.
पहले बता देते हैं मैम, वो तो आपकी
नहीं, उनको पता नहीं था, उनको पता नहीं था, उनको पता नहीं था.
मैंने जब देखा तो मैंने कहा कोई बात नहीं, हम तो फिर भी सर को थैंक्स बोल ही सकते हैं, और कहीं ना कहीं प्लेटफॉर्म. बट to tell him personally and to tell him with the audience that we have, he's such a well-recognized person.
To be frank, Dr. Uma, I was not knowing that you are so old timer क्योंकि उनकी student हो ये मुझे पता नहीं था.
मैं डॉक्टर मेहरा के साथ
क्या
PhD
बोलना
कर रही
चाह रहे
थी.
हैं?
No, when I was doing my PhD with Dr. Mehra, Dr. Malviya was my co-supervisor, and I did my thesis on spondyloarthropathies and HLA B27. So I used to go to his clinic to get the samples, and then my final doctoral committee meeting, when I was permitted to start writing my thesis, sir went to Gulf countries, Dr. Malviya.
Yeah, Qatar चले गए.
He was in India for that meeting. During the time we held the last meeting, that sir is in India. And once he, he told Dr. Mehra, okay, she has finished enough work. Let us permit her to write thesis. And then he called me and said, I will go, Uma, you can send me the thesis later. But if you want my signature elsewhere, you can take it now. And I was like surprised that such a senior person can be so humble and so concerned about a student that he said, I can sign it. And I said, sir, aapne thesis dekha nahi. He said, I trust that what Narinder will check will be fine and you can send me, I'll have a look. And he did have a look and responded to that. So it was such a, experience that such a senior professor can be so, so down to earth and humble. And wherever we meet, he's very, very kind and good human being.
Okay, nice
Yeah.
effort, Dr. Tarun.
Sir, thank you, sir. I think there are many who are still with us. I would like them to speak. Yeah, please. I think Dr. Krishna Prasad is there.
Prasad is there.
Yeah, and
Yeah.
we have. Hi,
How are you,
KP.
sir?
Hi,
Happy New
thank you,
Year.
Tarana.
Yeah,
I think it's wonderful to listen to Dr. Malviya.
Yeah.
And what I
Sir,
think
you
was very
can be on a
important.
video, sir.
Oh,
video
okay.
नहीं
Got
आ
to
रहा
see
है.
you.
All right.
It's a good opportunity for us to also meet again and again during these webinars.
Yeah. May I request everyone to please switch on their videos now? It is just an
Oh.
informal chat.
The video is not coming on.
No issues, KP.
Okay,
let us
I'm just
know
going to
what-
say, I thought one of the wonderful things about Dr. Malviya is like all of you referred to him as a great teacher. And I think it was wonderful to listen to him during the immunology workshops at AIMS.
Yeah.
You know, he and Dr. Ramesh Kumar, Dr. Talwar, Dr. Indira Nath, all of them, the whole lot of them were wonderful. And Dr. Malviya was outstanding for clinical immunology, and especially rheumatology, autoimmune diseases. I think it was wonderful to have him at that time. And I'm really happy that the Immunology Forum has been able to give him an opportunity and for all of us to listen to him also. Thank you.
Thank you, KP. Thank you for
sir.
joining.
To hear you.
I think Dr. Malviya sir was recognized by the society even at one of the immunocons, I think the Bombay Immunocon.
Mumbai. That's how I really came in contact with him, and that helped me to know him better.
Yeah.
Definitely. So, Dr. Anuradha, would you like to say something? Dr. Sunita Patil. Dr. Mary, you had a question. Unfortunately, I think Dr. Naik did not see the chat box and respond. I will pass on your question to Dr. Malviya, and I'll get back to you with the response.
Karuna, if you permit me, I need to leave.
Sure, Uma. Thank you so much, Uma.
Good to see you. Bye, Dr. Bala, Dr. Patil. Bye-bye, Foster.
Bye.
Patil sir, thank you.
Thank you, sir. Thank you, Balakrishna. Okay, Taruna.
Thank you, sir. Thank you so much.
Bye-bye.
Thank you. Thank you.
Thank you, sir. Dr. Bala, you have to inform me for the next one, right? Yes?
Yes, Taruna. The thing is, I
We'll talk on the phone. We'll talk on the phone, sir.
Yeah, okay, yeah, sure, yeah.
Okay.
Yeah. Okay, bye.